Hello and welcome back to another episode of AI 10X brought to you by 10 Leaves. Today we're taking a deep dive into the genesis of biotechnology. It's one of those fields that's constantly in the news with incredible breakthroughs happening all the time. But where did it all begin? What are the foundational stories that led us to where we are today? That's exactly what we're going to unpack. And to help us navigate this fascinating history, we have with us a true expert in the field, a historian of science specializing in the origins of biotechnology. Welcome to the deep dive.
It's a pleasure to be here. I'm looking forward to exploring the roots of this transformative science with you.
So to get us all on the same page, when we say biotechnology,
Yeah.
what's the essence of what we're talking about? It feels like it covers so much ground these days.
You're right, it is broad. But at its heart, biotechnology is really about us harnessing the power of biological systems, living organisms, or even just their tiny molecular components to create things that are useful to us. Think of it as applying our understanding of the natural world to solve practical problems and make our lives better. It's a very fundamental connection between biology and human innovation.
Okay, that makes sense. So if we think about this connection, this harnessing of biology, when did humans first start doing that in a meaningful way, even before we had the science to really understand what was going on under the hood?
Well, in a very real sense, the story begins with the dawn of agriculture, way back around 10,000 to 12,000 years ago. In places like the Fertile Crescent, early humans started selectively breeding crops and domesticating animals. They were essentially manipulating the genetic traits of these organisms through careful selection, even without knowing anything about DNA or genes as we understand them today. It was a form of biotechnology in its most ancient guise.
Wow, so those first farmers were unknowingly some of the earliest biotechnologists. Can you give us a specific example of how this early genetic manipulation through selection worked?
Absolutely. Consider the domestication of wild wheat. Early farmers would have noticed that certain wild wheat plants produced larger, more nutritious grains. They would have instinctively saved the seeds from those superior plants and planted them in the next season. Over countless generations, this repeated process of selecting and propagating the seeds from the most desirable plants gradually led to the modern wheat varieties we have today, which are far more productive than their wild ancestors. It's a testament to the power of observation and patient generation spanning experimentation.
That's an incredible thought. It wasn't a sudden invention, but this slow, deliberate partnership with nature over millennia, it gives you a whole new appreciation for the food on our table. But when we picture biotechnology today, we think of labs and high-tech equipment. When did that more modern chapter of the story really begin to unfold?
The shift towards what we consider modern biotechnology really began with some fundamental scientific breakthroughs that gave us the tools and the deep understanding to manipulate biology with much greater precision. A pivotal moment, of course, was the elucidation of the structure of DNA by James Watson and Francis Crick in 1953. Building on the absolutely crucial work of Rosalind Franklin and Maurice Wilkins.
The iconic double helix. We see pictures of it everywhere. But how did understanding this fundamental structure of life's blueprint actually revolutionize what we could do with biology?
Understanding the double helix was truly a watershed. It revealed the elegant mechanism by which genetic information is stored and passed on. It was like finally understanding the alphabet and grammar of life itself. This knowledge opened up entirely new avenues for understanding how genes function, how traits are inherited, and crucially, how we might be able to intervene at a molecular level to alter those processes. It moved biology from a descriptive science to one with the potential for directed manipulation.
Okay, so we had the blueprint. What were some of the first concrete ways that scientists started to actually use this newfound knowledge? What could they do that they couldn't before they understood DNA?
One of the earliest and most transformative applications came in the 1970s. With the development of recombinant DNA technology, this essentially gave us the ability to cut and paste DNA, to isolate specific genes from one organism and insert them into another, creating what we now know as genetically modified organisms. A prime example is the isolation of the human insulin gene and its insertion into bacteria.
And this is how we went from relying on animal sources for insulin, which I imagine was quite limited and had its own issues, to being able to produce human insulin on a large scale. That must have been an absolute revolution for people with diabetes.
Exactly. These modified bacteria became like miniature biological factories, churning out large quantities of human insulin. This dramatically improved the availability and purity of insulin for treating diabetes, a condition that was previously far more difficult and often fatal to manage. This technology also paved the way for the production of other critical biopharmaceuticals, such as human growth hormone, offering new treatments for various medical conditions. This really highlighted a fundamental shift from relying on limited natural resources to engineering biological systems to produce essential medicines.
So medicine was one major area, but biotechnology also started to have a big impact on how we grow our food right.
Absolutely. Biotech began to revolutionize agriculture as well. Scientists developed genetically modified crops that exhibited desirable traits like resistance to pests, which meant farmers could use fewer pesticides, leading to environmental benefits and potentially higher yields. We also saw the development of crops with enhanced nutritional content aimed at addressing malnutrition in different parts of the world.
Reduced pesticide use, higher yields, better nutrition. That sounds like it had the potential for massive positive impact on a global scale, especially with a growing world population.
Indeed. And the pace of innovation just kept accelerating. The development of techniques like polymerase chain reaction, or PCR, and gene sequencing were game changers. PCR allowed us to make millions of copies of a specific DNA sequence from even a tiny starting amount, while gene sequencing gave us the ability to read the entire genetic code of an organism.
You mentioned PCR. That's a term we hear quite a bit, especially recently. Can you give us a quick, easy-to-understand explanation of how PCR works and why it was such a big deal?
Think of PCR as a molecular photocopying machine. It allows scientists to take a very small sample of DNA and, through a series of heating and cooling cycles with the help of special enzymes, create millions, even billions of identical copies of a specific DNA segment. This ability to amplify DNA is incredibly powerful because it allows us to work with minute amounts of genetic material, which is often the case in research forensics and diagnostics. It really democratized and accelerated biological research, making it possible to analyze DNA evidence at crime scenes or detect diseases much earlier and more reliably.
A molecular photocopier. That's a great way to visualize it. And the ability to read the entire genetic code through sequencing must have opened up even more possibilities.
Absolutely. Gene sequencing provided an unprecedented level of detail about an organism's biology. It allowed us to identify genes associated with specific diseases, understand genetic diversity, and trace evolutionary relationships. This knowledge laid the foundation for advancements in personalized medicine, where treatments can be tailored to an individual's unique genetic makeup.
It's amazing to think about how these foundational discoveries built on each other, each one unlocking new possibilities. Were there other key figures beyond Watson and Crick who really pushed the boundaries of what biotech could achieve in those early days?
Absolutely. Rosalind Franklin's X-ray diffraction images were absolutely crucial for Watson and Crick's breakthrough, providing the essential structural information about DNA, although her contribution was not fully recognized during her lifetime. Then there's Kary Mullis, who invented PCR, a technique that underpins so much of modern biology, and Barbara McClintock, whose groundbreaking work on transposable elements, which she called jumping genes, revolutionized our understanding of the genome. She showed that our genetic code isn't as static as we once thought. Genes can actually move around within chromosomes, which has significant implications for gene regulation and evolution. These are just a few examples of the many brilliant minds that contributed to the early development of biotechnology, each building on the work of those who came before.
It really sounds like it was a period of incredible intellectual ferment, with these amazing discoveries constantly reshaping our understanding of life itself.
It was, and those early discoveries provided the essential groundwork for the truly extraordinary advancements we're seeing today. Think about CRISPR gene editing, which allows us to precisely alter DNA sequences, or synthetic biology, where we're essentially designing and building new biological systems. These are fields that were almost unimaginable just a few decades ago. And they all have their roots in those foundational discoveries we've been discussing.
It truly is mind-boggling how far we've come in such a relatively short time. This has been an absolutely illuminating journey through the genesis of biotechnology. It's fascinating to see how these early steps, from selective breeding to understanding the structure of DNA, laid the foundation for the incredible advancements we're witnessing today. Thank you so much for sharing your expertise with us.
It was my pleasure. Thank you for having me.
Stay tuned for more insights on AI. This is AI 10X, a podcast series brought to you by 10 Leaves.